Hello dear students of the first secondary grade, and welcome to a new episode of our program. On this episode we're going to study some biology, and we're going to continue the lesson we started on our previous episode. It's chapter 2 of unit 3. We're going to talk about the interaction of genes and study how to determine the blood groups in a human being with our teacher of biology, Ms. Violet Khayri. Hello, Ms. Violet, and
Hello.
welcome to the program.
Thank you.
So let's first start by where we stopped last time.
Okay. Last time, my dear students, we started studying the blood groups and how the blood groups genes are inherited from one person or one generation to another. And we studied also the importance of blood groups and the blood transfusion. And today we're going to complete what we've started last time, which is about the determination of blood groups. How to determine the blood groups in men. Actually, my dear students, as you studied, that the blood groups is determined because of the presence of certain gene that causes the appearance of antigens on the red blood cells, and these antigens of many types, as we said last time. The blood group A produces antigens A, the blood group B produces antigens B, and the blood group AB produces antigens A and B, and the blood group O has no antigens at all on the red blood cells. So the blood groups A, for example, produce in the serum or in the plasma antibodies against the different antigen. So the blood group A produces antibodies against the blood group B. The blood group B produces antibodies against A. Okay, so this is very important to determine the blood groups. In this procedure, we started by having 2 drops of blood from the person and put them on a glass slide and add a drop of the antibodies produced or extracted from the serum of the blood group. So the first the first drop from anti-A, and the second drop from blood group A, which contains anti-B. Actually, if there is a different blood group, so the antigen A or the antigen A present in the red blood cell meets the antibodies A, there must be a reaction between them, and the antibodies cause the agglutination of the red blood cells and destruction of them. As you see in this picture, we put anti-A on the first drop, and as you see, there is a chemical reaction. So the blood is group A, because in the same time when we added the antibodies or the serum containing antibodies on the second drop, there is no agglutination. So this is blood group A. So if reaction occurs with the blood and the anti-A, so the blood group is A. But the reaction occurs between the blood group, the blood B, with anti-B, so this is group B. And in the same time doesn't cause any agglutination with the anti-A. But if reaction occurs between the serum of anti-A and anti-B, so this person is AB group. But if no reactions occur between the anti-A and the drop of the blood and anti-B with the blood, so there is no agglutination in both cases, so this is blood group B. So systematically, you can guess the reaction with anti-A, so the blood group is A. The reaction with anti-B, the blood group is B. The reaction with both is AB. No reactions at all is group O. The risks of blood transfusion. When an incompatible blood with the blood group, so if we make transfusion between two different blood groups, the person is transfused to him. This includes symptoms such as shiver, headache, chest pain, breathlessness, and many other complications that lead finally to death. So it must be of the same blood group. Now we come to another part of studying the blood, which is called rhesus factor. What is the rhesus factor? The rhesus factor is an antigen, another antigen present on the red blood cells, and it was discovered first in the monkeys species called rhesus, so we call it rhesus factor. So there is another type of antigens on the surface of red blood cells known as rhesus factor. Actually, this character is controlled by 3 pairs of genes, and these 3 pairs of genes locate on 1 homologous chromosomes. Have a look with me. So this is 1 homologous chromosomes, for example, and there are 3 pairs of genes on the same homologous chromosome. This is pair number 1, pair number 2, pair number 3. And it follows Mendel's laws that dominant and recessive. So any one of these 6 genes is present in dominant state, so the person is called Rh positive. So we have 6 genes or 3 pairs of genes. Any one of them is present in a dominant state makes the person Rh positive. But when this person has no dominant gene, so the person could be or must be Rh negative. Okay? This makes the most of people are positive, Rh positive, because we have 6 genes. So about 85% of human beings are Rh positive, and only 15% of the human beings are Rh negative. What is the importance of rhesus factor or studying rhesus factor? Before blood transfusion, so we need to know whether the person is positive or negative. Otherwise, it causes many complications and lead to death. So there must be known, it must be known that whether the person is positive or negative before having the blood transfusion. Also, it is important before marriage to avoid risks when it causes disintegration of red blood cells. Let us have an example here. If a man with blood group Rh, the blood is Rh positive, and the woman is Rh negative, what happens here? The man gives his child the Rh positive gene, and because it is dominant, so even the presence of Rh negative coming from the mother, the Rh negative gene coming from the mother, he is Rh positive. So what happens here? My dear students, bear in your mind that the person who has Rh positive and has blood transfusion Rh negative, the blood does not have the ability to produce the antibodies against the Rh negative. But the person who has Rh negative has the ability to produce the antibodies against the positive one. And this is why it is very dangerous for the person who is Rh negative to have blood positive. And this occurs during the pregnancy. The role of rhesus factor in pregnancy and delivery. What happens when an Rh positive man married an Rh negative woman? During this case, during the pregnancy, the fetus has a separate blood circulation, and the mother, of course, has a separate blood circulation. But they meet at the placenta. Or in the placenta, the blood vessels from each side is very close to the other one, and the blood, the diffusion of materials coming from the mother, like food, oxygen, and vitamins, goes to the fetus, and the waste materials coming from the fetus to the mother. But during child delivery, the placenta is ruptured, and the blood may transfuse or goes to the mother. The blood of the fetus may go to the mother and causes the production of antibodies against the Rh positive. And if the antibodies coming from the mother to the fetus comes from the mother to the fetus, these antibodies may cause the disintegration of the blood of the fetus and may cause death. But actually, it doesn't occur in the first baby or the first child delivery, because he is born before the formation of these blood groups. But for the second baby, the mother has already in her blood antibodies against the Rh positive. And of course, the baby is Rh positive, so the antibodies goes from the mother to the child or the fetus and causes death to this fetus. So the antibodies move from the mother's blood to the fetus's blood through the placenta during the second child. How to prevent this disaster? To prevent that, the mother should be injected with a protective serum within 72 hours after each birth to prevent the mother's immune system to form the antibodies. So after each delivery, the mother should be injected with this protective serum to prevent the formation of the antibodies, and the next fetus will be safe from death. Now, my dear students, we come to another method of gene interaction, or another case. This is called complementary genes. You know that Mendel worked on edible pea plant, but now we are going to study the garden pea plant or ornamental pea plant. In ornamental pea plants, the scientists noticed that when two different white flowered plants were crossed together, the produced individuals in the first generation were pink. And it's not usual, it's very strange. And by crossing the first generation, or allowing them to have self-pollination, or crossing them with each other, the second generation will be pink and white. Let's see this case in details. The genes that can work together to emerge a specific trait. This is what we mean by complementary genes. So the word complementary means the genes complete the work of each other. They work together. Each gene produces or causes the production of certain protein, and the two genes or the two proteins produced complete the action of each other to produce the color of the flower. Otherwise, if one of them is not in a dominant state, so the other gene produces its protein, but it's not enough to produce the color of the flower. So there must be the two genes are dominant state even if they are hybrid. They are controlled by two pairs of genes. The emergence of a dominant character depends on the presence of a dominant gene at least in each pair, as we said. The recessive character appears only when one of the genes or both are recessive. How can we explain it in details? Yes. So if we have the parents, white flowered plants, both are white flowered plants, and we get in the first generation pink flowered plants. As we said, this type of inheritance or these characters are controlled by two pairs of genes. So the first pair, if the first pair is white, so there must be one of the two genes present in a recessive state. So if we have A for the first gene and B from the second pair of genes, this is white flowered plant, because the first gene produces the protein and the other pair doesn't produce its protein. So this means that this the proteins coming from this pair does not find the complementary protein to produce the color. So the plant will be white flowered. And the other one is the same. We assume it is a small, and capital B. Also, this individual has the first pair in recessive state and the second pair is in dominant state. So it will not be colored, it will be white. What about the gametes? Remember, the gamete is a cell, reproductive cell, contains half the number of chromosomes. We take one half from the first pair and one half from the second pair. This is the first gamete, and also one half of each pair. So these are the gametes that we have. After pollination and fertilization, the seeds are formed, and when we cultivate the seeds, all of them were pink. So as we see here, A capital with A small, B capital with B small. Why they are pink? Because in the first pair there is a dominant gene, and in the second pair is a dominant gene as well. So both complete the function of each other to produce the pink color. But what happens when we allow the individuals to have the self-pollination or even cross-pollination, because all of them have the same genotype? A capital A small, capital B small b. These are the parents of the second generation. And the gametes, okay, as we said, we have two pairs of genes. We will have half of them half of each one. The half of the first pair and half of the second pair. But which one? Yes, it goes with the law of probability. So it could be the gene with the chromosome with a capital or dominant gene A with the chromosome with the dominant gene B. So A comes with B, A capital with B capital, or A capital with B small, or A small, B capital, or A small, B small. As I say, or, not and, because we put the types of gametes, the probability of the gametes, not the number of gametes. And these are the gametes of the first individual. What about the second individual? Yes, it's the same. We put them in a table as you used to do, like this. And let us follow in the screen. And these are the parents, two white flowered plants, and the gametes to produce the pink flowered plants. These are the individuals of the second generation, and these are the gametes of first individuals, gametes of second individual, and by crossing the gametes with each other, we will find that some individuals were pink and others were white. By counting the number of the individuals with pink flowers, they are 9. But what about white flowers? Yes, 7. What are the genotypes, possible genotypes of pink flowered plants? It could be capital A, capital A, capital B, capital B, or capital A, small a, capital B, capital B, capital A, small a, capital B, small b. So these are the individuals, capital A, capital A, capital A, small a, capital B, small b, as we said here. So these are the individuals that could be pink. What about the white? There must be one of the two parents in recessive state. It could be like this. Yes. So one of them is recessive or both are recessive. Sorry, small b, small b. So these are the genotypes of pink or white flowered plants. Okay, so the ratio here is 9:7. And remember, in Mendel's second law, we said that this 7, in the second law, we were studying two pairs of genes with two different characters. And we said that the 3-3-1 means the first pair is dominant and the second is recessive, or the first pair is recessive and the second one is dominant, or both are recessive genes. So my dear students, we have to make a comparison between the complementary genes and the Mendel's second law, that is law of independent assortment, and of course we will discuss it later.
Yeah, can we do this next time? All right, so I'd like to thank you very much, Miss Violet Khayri, for this episode. Thank you very much. So dear students of the first secondary grade, we've been with you for almost a half hour, and we discussed the interaction of genes. So we talked about the blood groups and how to determine blood groups in man. Thank you for being with us, and until we meet you again, wish you all the best. Goodbye.
Thank you. Bye-bye.